It is officially past 11 Eastern, 8 a.m. Pacific. So good morning, good afternoon, good evening, everyone joining us from around the tinyML community here. So we're going to get started now with our talk. Really looking forward to the agenda that we have today. Not two, but three tinyML talks that are going to be really exciting, cover a broad range of topics. So without further ado, let's get started here. So my name is Davis Sawyer. I'm one of the co-founders of a tinyML company called DeepLight and the local chair for tinyML Montreal and also Toronto. We have a really burgeoning tinyML community here in Canada, so we're really excited to be able to share some local and also some speakers from abroad. Also, the community is joining us from Parker Zhang from Qualcomm and Kevin Favre of Toronto, LG Toronto. So really excited to be able to put this together for you today. First of all, I'd like to thank our tinyML Talk strategic partners, Aeon Devices, ARM, DeepLight, Edge Impulse, Emza Visual Sense, Greenwaves Technologies, Gravity Inc., HotG, Imagimob, Clickatek, Latent AI, Maxim Integrated, Micro AI, NXP, Prophecy, Qualcomm, Kixo, Reality AI, Renaissance, Rixen, SAP, Seed, SensiML, ST Micro, Stream Analyze, SynSense, and Sentient. There are additional sponsorships available, so if you're interested and want to join the tinyML community, get in touch with Olga. You can see her email here below. A couple of really exciting things, actually. So we're already kind of mid-January. We do have the tinyML Summit 2022. This will be an in-person event at the same location it was in the past at the Hyatt Regency San Francisco Airport, March 28th to 30th. Registrations will open this month, so stay tuned for that. And this will be an in-person event, which is super exciting. And concurrently, there is the tinyML Research Symposium. This is an open event. This is really a great chance to get up to speed and share what's happening in the research community here. So that will also be held at end of March. And again, sponsorships are available here. So in parallel, there is the growing tinyML communities on Meetup. You can join the link here below. As I mentioned, this is our first virtual chat from the Toronto group. Always like everyone staying tuned for when we can do more of the in-person events. But tinyML is a global organization, can join here for anyone who's new. And then there's also the LinkedIn community as well. So also there is the YouTube channel. So all the talks that's from today and the talks from the past, all go to the tinyML YouTube channel. This is an awesome catalog and resource of all the different topics that have been spoken about. And you can find that here at the YouTube channel. And the next tinyML talk, it will be just around the corner, January 18th, with Ashutosh Pandey from Infineon Technologies. The topic will be exploring techniques to build efficient and robust tinyML deployments. So that will be same time as usual, 8 a.m. Pacific on the 18th. We have Greg Gage from Backyard Brains. Greg is a neuroscientist, engineer, and the CEO of Backyard Brains and helps lead the tinyML4STEM effort. Greg develops tools, curriculum, and experiments that allow the general public to participate hands-on in neural discovery. He is a senior fellow at TED and has given many TED Talks. 9 of them are online. He received the Director's Innovation Award as an investigator at the National Institute of Health and was recognized in a White House ceremony for being Obama's champion of change for his commitment to citizen science. In his free time, he enjoys changing diapers of his 2 young daughters. Awesome. Thank you, Greg, for taking the time today.
My name is Greg Gage. I'm a neuroscientist. And so what does that mean is I study the brain. Let's see if I can get this to work. And so if you're not familiar with how neuroscience is typically done, we have large racks of expensive equipment and we study the brains of animals during their behaviors and this type of stuff. And it's unfortunately a field where people feel that you have to be at a major university with a lot of funding in order to be able to do research on this. And that's a shame because 1 out of 5 people, that's 20% of the entire world, has a neurological disorder. And let me see if I can move this up. There are no cures for any neurological disease that's out there. So it's a bit of a shame that the only chance to get to study the brain and to learn about the brain is to dedicate your life and become a neuroscientist that slaves away for 6 or 7 years in your PI's lab just to get access to these tools. And so when I was in grad school, my lab mate and I decided that there's a better solution, and that is to develop some solutions that can democratize neuroscience and bring it into the classrooms and to get more people interested in neuroscience. You can look at For example, as an analogy is in astronomy, you don't have to get a PhD in astrophysics to look through a telescope, right? And a lot of people do look through telescopes that are amateurs, and they make major discoveries, exoplanets, the Hale-Bopp comet. So there's a potential that we could actually expand the amount of research being done on the brain by sort of bringing it out to the people. And so we've been doing this for about a decade now. We have a series of lab protocols that we're putting up on our website. Each one is kind of peer reviewed through the, put out in the scientific literature. And most of what we've been doing is on biology, but I've been, for the past 9 years, I've been teaching a computational neuroscience course at Woods Hole. And we have yet to come up with a cool way to democratize, AI and machine learning in the same ways that we've been able to do it with our hands-on kits for doing physiology. But there's a lot of reasons why that's important for a neuroscientist to be sort of talking about AI, because as you're quite familiar, the entire modeling system came from neuroscience, right? The idea of these synapses coming from cells to cells. You got nodes that are connected together in a different mesh. And so we have been struggling to come up with a way to make this a bit more hands-on. Every lab we've had so far has just been like students sitting on a computer, and they can classify irises, but I feel like they walk away feeling a little bit like they understood what's going on, but not quite really. And so with the advent of tinyML, that was kind of the light bulb that was like, all right, now we can start bringing this into the classroom and have people really start to understand how this AI is working by collecting their own data sets. And so we have a pretty rich environment of data to collect from, which is cool. And it's also very personal to the students, right? So we're going to be looking at sort of what does the electricity in the body sort of give out that we could pick up on sensors and feed it into tinyML. And so really what we've invented are what's called bioamplifiers. In science, many things are invented to make things bigger, right? So you need, telescopes to make distant planets bigger so you can see them. You need microscopes to make small cells bigger so you can see it. PCR machines make DNA bigger. What these things do, these are little bioamplifiers that make little tiny voltages of electricity big enough so that you can, see it and hear it. And so that's what our sort of our inventions are around this idea. And so what I'm going to talk about today are what are the signals that are good for tinyML, and just give a little bit of a background on some of these things and how some are better than others. The first one is your electromyogram, or EMGs. And this is I like this one. It's kind of the hello world of physiology, and I think for any type of embedded computing system. So just some background: neurons fire electrical impulses throughout your cortex. And I will shout out again. I think Lang brought this up, but this is a really good book. If anyone has not read this yet, it's a good theoretical background on how cortex works, and I think it has a lot of cool implications for the future of AI. But anyway, your cortex has these neurons that fire in layer 5. They project down to your spinal cord in your motor cortex. And then from your motor cortex, it goes, from your spinal cord, it goes out to your muscles. And if you put some electrodes on your muscles, you have a little bit of salt water there. Then what you can do is you can connect those to some wires here. And the wires are metal, right? So electricity will flow down the wires and go into our amplifier, and then go into our Arduino kits that you'll be able to run some tinyML on. I'll give a quick example of what that sounds like. I'm going to turn on my amplifier here. You guys can hear that. So you're recording those neurons inside the basically from the spinal cord is projecting onto the muscle fiber, and you're able to sort of hear that. And if we are careful, we can actually see what the signal looks like. So here's the actual signal itself is a spike. Let's see if I can find. I have to move this thing out. I'll show that right now. But anyway, so the signals themselves are little spikes they're kind of being compressed onto each other. But what's cool about this is a 1D signal. And so it's really easy to stick into, for example, an Arduino to make it do things. So here's a classic Arduino, and I'll show you what we're going to be doing with tinyML first. The first thing we do in physical computing is make it do like a hello world of getting to turn things on, LEDs on and off. You go a little bit more advanced, and you can make a little neural prosthetic, a brain machine interface. So you can have a hand that closes when you close. So this type of stuff. It's a beautiful, like I said, one-dimensional signal that it's really easy to then stick into tinyML. And so what are we doing with tinyML here? We have the same type of idea, but now with doing some inferences on which fingers are being moved. You can have robotic hands sort of copy that on. And I'm going to talk a bit more about some other signals other than EMG. EMG is the easiest one to get access and get up and running with. The next one will be the electrooculogram. So your eye is a dipole, so it's like a little battery, and so as it moves around, if you have sensors on there, you can detect the position of the eye. And so what we were doing is with the EEGs, we're not detecting the actual position of the eye until if you see, I've got one above and one beneath, and so you can see that red line, she was looking up. So you get this DC signal that kind of just moves around. But you see these green lines are indicating something else that's happening. And that's an eye blink. And so we can detect that one of the fastest movements in the body is your eye blink. It lasts, less than, I think it's around 200 milliseconds. So it's really, really fast. And so, but you don't even notice it. Your eye kind of, your brain sort of blanks all that out and merges together. But what is happening when you are blinking and you don't see it? So at the end of the day, you're going to be able to miss out. And actually, the end of your life, you live to be about 80 years old. I think 2 years of your life are given away just to your eyes being closed when you were blinking, right? So we are going to recover that using tinyML. So what we've done is trained a network to detect the eye blinks and reject all the other movements, artifacts, and stuff like that you're getting from the EEG. And then we made a set of glasses that just took the tinyML kit from Arduino and just repositioned the things on a set of glasses with an EEG sensor on there. So you're able to take a picture every time the eye blink gets detected. And at the end of the day, you can watch a little movie of what you missed out on. So we call them FOMO glasses. And so you will never be able to, miss out again on what's going on. So another example is the EKG, electrocardiogram, or ECG in the US. And so this is where your heart's pumping. And so why is that important for neuroscience is that you have your autonomic nervous system, which has the sympathetic and the parasympathetic nervous system. And one of the ways that your body, this is like if you're looking at sort of like the older part of the brain communicates to the cortex through this system, right? And so we have ways that we can determine our subconscious is afraid, or our subconscious, like this type of stuff gets fed back to the cortex in the form of these signals, right? And one of them is your heart either accelerates or slows down. And so it's a way that you can sort of detect some involuntary things that are going inside the brain. And so for example, if you were to put your hand in ice water as a stressful event, you can just easily see your EKG start to increase. And so what is that useful within tinyML? So you can do things like we had a student this summer working on a skin galvanometer. So that's the same. It's another signal that comes from the autonomic nervous system. So you get the sweat on your skin, and your EKG dramatically change when you're stressed, right? And so we had a student that played poker, and so he was training tinyML on when the river turns, what was the response from the person, and did it turn out to be a bluff or not? So he was detecting if it was a bluff or not based upon these biosignals that are coming back from the human subjects. And then I want to talk a little bit more deeper about EEGs. There's a lot of misconceptions about what the electroencephalogram is, how much information is there. I will tell you there is not a lot of information in the EEG. There's some, and some useful stuff that we can talk about in just briefly, but I have a book that's coming out this fall, and these are some images from there. But the idea is that I just want to stress that you can't read your brain from an EEG. I think there's a lot of misinformation that. So here's an example of someone thinking about a dog and a cat and, a stressful exam or relaxing on the beach. And the EEG signals look exactly the same. It's going to be hard for you to pinpoint one thing or the other. And so you can also look at the intensity of the signal. If you really think about something really blue, and then you kind of think about something that's a little bit blue, it's not going to change your EEG at all. It's all going to look the same. So don't worry about having a brain scan or reading at least your electroencephalogram for this. But I just want to talk a little bit about what is the EEG and how does it work, and then what can we do about that. And so what your neurons are doing, each neuron is sort of doing predictions, but each one is trying to predict a different aspect of the sensor information that's coming in. So you can have, if you're looking at the visual cortex, I'm looking all around. So I have little neurons that are responding to different pixels that are on the screen and background stuff. So it's really, really complex. And so, but versus if you have everything that's kind of working together at the same time, then you can start to get a signal to work. And I have a quick analogy of that. And the analogy would be if you're sitting outside of a ballpark and you're trying to record a conversation that's going on inside the ballpark from the outside, you're not going to be able to pick up anything, or you're going to get a whole bunch of noise of people just kind of, just sounding like a dim of a crowd noise. That doesn't mean you can't get any information out from that, right? So, for example, if they're starting to sing the national anthem, that the game's about to begin because you can recognize that because everyone's singing in unison, and you start to get a signal out, and you can start to detect that, right? So, this is again a really good analogy for EEGs. When groups of cells are doing all the same thing at the same time, then you get to see something like this, right? And so. The other thing is like if you can tell if there's a home run, if everyone's cheering at once or stuff like that, or some sections are cheering, and you can tell a wave is going around just by listening to these sounds, right? So these are the type of experiments you can do. So if you think you can record from a single neuron activity inside, you're not going to have much luck. You're not going to be able to get much information from that. And so what do the signals look like that come out of the EEG? They're really kind of two ways that we look at them. And so one is a wave signal. So it's like these are oscillations that occur. And the other ones are event-related potentials. And so when something happens in the outside world, for example, like that turning of a card would be an event. And then you can look at the next X number of seconds, you can pick up information that way. So these are the two ways that EEG are used. And I'm going to talk about how we can use them within tinyML. So here's an example of that idea of that when your eyes are open, you're seeing a whole bunch of information. And so therefore all the neurons are doing different things. But when you close your eyes, all the neurons are doing the same thing. They're in this kind of repeating pattern, waiting for the next input. And so if you put some electrodes just on the back of your head, you can see this in the terms of an oscillation. This is the EOG artifact. That's that blink artifact we're trying to detect in the EOG. But here in the EEG, you start to see these 10 hertz oscillation, this alpha wave that appears above the visual cortex when you close your eyes. Okay. The other way we can do it is with these event-related potentials, and these become important for tinyML as well. And so this is called the P300 task. It's really cool. So this is used in hospitals to see if you're brain dead. So if you get, if you play a series of tones and then all of a sudden you put an oddball tone in, so beep, 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 boop, beep, beep, beep. You can look at what the brain signal is like during the beeps and the boops. And the one you're not predicting to happen, which is called the oddball tone, you get this bump up of activity in the EEG in a certain part of the brain right after that occurs. And so it's really easy to sort of train a tinyML thing, given the event marker, to give this section of the EEG to determine if it was a beep or a boop. And so another example is using the frequency stuff. If you flicker something on the card, this is used for people who have locked-in syndrome, or people have a brainstem stroke that can't speak anymore. They can still use their eyes to move their eyes around on the screen, so they can look at different things. And if you flash letters, or this is the example where we're having them pick cards, like a magic trick. This is done on a little seed studio tinyML kit. So we're flashing the cards at different frequencies, and if you stare at the card you can pick up in the EEG the frequency at which that card is flashing in the visual cortex, and so you can so we have 4 windows at which you're looking at, so you can tell which card the person is looking at just by doing some training on those segments of the EEG. So, and I want to talk about one more, which is kind of cool, and this has to do with the event-related potentials. So it turns out we may not have the free will that you think we do. This is it was actually a controversial thing in the 80s. So this is the idea that that when you saw my arm moving, you saw the EMG on the screen. So, but if you train if you record from your motor cortex in the EEG and you trigger it on when your EMG appears on the screen, and you keep averaging it over and over again every time your EMG, your movement onset occurs. So that's where this red line is right here. But you look back at what the EEG was doing before that happened, right? And so it turns out that, almost this is over a half a second before you move your arm, the EEG signal starts to come up significantly. So it's something that we could detect, right? And then you ask the person, well, when did you decide to move your arm? And they say, well, I just decided to move it. And then you can actually put a measurement clock on there, and you can figure out when did they decide to move the arm. They decide about 150 milliseconds before they move it. However, we can detect with the EEG 500 milliseconds before you move it. So there's a period of 400 milliseconds, hopefully, that a computer can detect you're about to move your arm before you're even aware that you are about to move your arm. So again, these are concepts that are referenced in that book, The Thousand Brain Theories. It kind of makes perfect sense because your subconscious is sort of feeding this information into your conscious thought. You think you come up with the idea, but it's that in fact it's the whole brain working together to do that. But anyway, so we're trying to do this right now. We're trying to make a machine that can detect using tinyML to detect when you're about to move before you move. And then we're going to have this experiment where you have a light that comes on and you can move your hand whenever you want, except for when that light comes on, right? And so then we're going to see how well we can predict it and have the light come on before you're about to move your hand, and then kind of mess with you. So that's kind of the goal of what we're trying to do. So if you like this type of stuff, you feel free to stop by our website. We're at backyardbrains.com. We have we're just getting started on the tinyML stuff. So if you have ideas or you want to participate in some of these projects, we'd love to work with you. So with that, I'm going to open up for questions, and thank you guys for your time.
Awesome. Thank Greg. That was great. I think we had a lot in today, with the speakers, but that was a great way to wrap things up. We're going to try and quickly get a couple questions here, and then we'll do, the wrap up here. So Greg, first question is, I can imagine that the position of the sensors highly impacts the accuracy of model inference. How do you deal with that?
Yeah, so we haven't quite figured that quite out yet. So we have noticed that with the EMG, you can do the positions of the sensors will change dramatically because you're placing it right over these muscles. And so how we're going to solve that is by doing just some quick tune-ups when you first get going. And so we're going to have 2 modes, a training mode on the device itself in the code that they'll switch back over, so then you can start using the inferences from that. The next will be and the EEG. All these other ones don't really matter. So pretty much everything else, the EEG is such a diffuse signal. It doesn't really matter where you're placing that. You have to be. It's kind of like horseshoes and hand grenades. You get close enough, and you're going to get a good signal. And so then that becomes an issue of training. If you just get a nice a training set with the electrodes in different locations, then you should be able to cover that one. So. A bit. The EOG is the same thing. So most of the ones that really matter are like the EMGs, but then you can just use large muscles that it doesn't really matter where your things are, right? So that's the other thing. So if you want to do fine motor movements, then it really matters. If you're doing gross motor movements, arm movements, stuff like that, then not so much. And so you can just solve that by training over a larger data set.
Makes sense. Cool. And the last question here, how many channels of signal did you use in your Arduino demo with moving fingers? Was it only one wire signal, channel signal?
Yeah, so that was originally, I think we had 5. I mean, it was kind of a easy task for tinyML, right? So you had 5 signals and 5 unknowns, right? So, but what we'd wanted to do, we haven't gotten that far, is to see how far we can push it. Like, can you use 2 electrodes to be able to do 5 figures. And so I suspect you should be able to do much, much more than, or much less than 5. But the student's project was done at the end. He was able, I didn't show the video, but yeah, we were able to do it pretty quickly. Be able to infer the finger positions. But I suspect, 2 should be able to do it.
Gotcha. Awesome. Well, so on that note, thank you, Greg. That was a really interesting presentation. Shows off, I think, what tinyML can do. And on behalf of the tinyML Toronto group, thank you to all the speakers and everyone attending today for kicking things off. And thank you to all the strategic partners. And again, there's also additional sponsorships available. First of all, so the executive strategic partners of tinyML, you have ARM, the software and hardware foundation for tinyML, Edge Impulse, the leading EdgeML platform, Qualcomm AI research, advancing AI research to make more efficient AI ubiquitous, sentient neural decision processors, end-to-end deep learning solutions for tinyML and Edge AI, and to the platinum strategic partners, DeepLight uses AI to make other AI faster, smaller, more power efficient, from software to runtimes. Clicatech Global IoT Solutions. Reality AI, add advanced sensing to your product with Edge AI tinyML. Renaissance, broad and scalable edge computing portfolio. And to the gold strategic partners, Maxim Integrated, enabling edge intelligence. Latent AI, adaptive AI for the intelligent edge. Also Micro AI, NXP Semiconductors, Seed Studio, the IoT hardware enabler. SensiML builds smart IoT sensor devices from data. ST Micro, life augmented. SynSense builds sensing and inference hardware for ultra low power embedded mobile and edge devices. And to the silver strategic partners, Aeon Devices, Emza Visual Sense, Greenwaves Technologies, Gravity Inc., Hotg, Imagimob, Kixo, Prophecy, Recent Technology, SAP, and Stream Analyze. So the next talk, again, right around the corner on the 18th, 8 a.m. Pacific, 11 Eastern, we have Ashutosh Pandey from Infineon Technologies, talking about robust tinyML deployments. And with that, I'd like to thank everyone for making this a really fun, really successful first talk from tinyML Toronto. Keep in touch for more groups getting involved in the community, joining the meetup groups, YouTube. Everything's available online. Thanks again to the speakers, and wishing everyone a great day.